All right, good morning, everybody. Let's see where we're at today. Happy Friday too. Woohoo. The weekend. All right, get everything up. Dum-de-dum-de-dum. All right, so we are beginning on gene expression. All right, this is a big one. And as I was telling the other class yesterday, so you've had a taste of, this kind of molecular genetics already, right, with DNA replication. And you saw kind of what that was like in exam 2. So the rest of the semester will be like that. There's going to be a lot of details, a lot of kind of processes with details to, kind of flesh them out. A lot of kind of abstract stuff, right? So obviously all of this underpins everything biological, right? It's not, I can't think of anything, no matter how kind of big a scale or, ecology or evolutionary focused you might, be interested in. There's nothing that is not impacted, biology wise at least, by gene expression and its control, right? So it is very much, it's super important. Obviously, that's why we spend a whole bunch of time on this. But also the downside is it's kind of abstract, right? It's, molecular biology and molecular genetics is, kind of stuff you learn, and I can integrate that into kind of bigger picture stuff, but it's not quite so easy for you lot. So it can be really hard to learn. That's kind of where I'm getting at. So you really need to kind of apply all of the strategies that worked well for you so far, right? And the exams are actually doing pretty okay, to be honest, a bit better than prior semesters. But keep on that, and like even more so, right? So figuring out what you don't know, because a lot of this stuff is going to make sense when I'm telling it to you, but I'm not going to be there in the exam to help you, right? So, well, at least answer the questions. So you really need to figure out what bits of this stuff you don't understand before you get in the exam, right? So, testing each other, quizzing each other, working in study groups, getting your family, kids, parents, spouse, whatever, to quiz you on stuff, right? Being able to write out these processes kind of by hand without reference to any other material, right? So it's purely stuff that's in here. All of those things will show you what you don't know, right? And that's what you need to study. And this is, some people get on really well with this stuff because that's just kind of way their brains are wired. I mean, that's kind of me, for example. But a large chunk of people find this stuff difficult, right? So just kind of giving you the heads up because it's going to be a pretty intense, in-depth kind of look at these mechanisms. All right, so what we're going to do is we're going to look at kind of principles in general first. It's kind of what we, usually do actually. Things that are shared amongst all life in terms of gene expression. And then we're going to look at some particular quirks of prokaryotic gene expression. And they have some, funky tricks up their sleeve. And also then we're going to go on to eukaryotes, and eukaryotes are quite a bit more complex in many ways. It's kind of a common theme, to be honest. And one of the reasons why that is so is because of the structure that you're seeing in the picture in front of you. So eukaryotes have a nucleus. It's one of the defining features of a eukaryotic cell or organism, right? It has a nuclear membrane, this thing here, which separates the DNA from the cytoplasm, right? And so DNA is in the nucleus, protein is produced in the cytoplasm, mRNA is the thing which kind of bridges that gap. right? But there's a lot of stuff that needs doing to mRNA before it can, be released into the cytoplasm. So that's one of the many quirks of eukaryotic gene expression. Okay, so by definition, really, gene expression is a production of whatever the things, proteins or RNA typically, that give you the phenotype that you have for a particular trait. It's whether or not a gene is expressed, what form that protein is in, whether it's functional or not, and, where, when, how much, that kind of stuff. So I always use alcohol for some reason. I'm not much of an alcohol drinker, but great example is alcohol tolerance, right? So I have none. Well, a little bit. Cheap date, basically, because I don't drink very much, right? If you know someone who drinks a lot, then they can drink a lot and still be functional, right? And the reason for that is that they are constantly producing a variety of enzymes in their liver, including one called alcohol dehydrogenase, which detoxifies ethanol, because ethanol is actually a poison for us. Hey, quit it. Cat scratching at the wall. And people that don't, right, you don't need that protein, right? So protein, gene expression is costly, right? So there's a benefit to not doing it if you don't need it. There she is. Bloody pest of a cat she is. Don't you? Yeah. Comes and yells at me. Actually, she yells at everybody at different times of the day. And so, I don't produce much alcohol dehydrogenase because I don't drink much alcohol. So protein is the thing that does stuff, by and large. So protein is a sequence of amino acids, right? So if you want a kind of definition, it's a sequence of amino acids joined together in a particular order to make a polypeptide. Pardon me. That is then processed, obviously depending on whether it's eukaryotic or prokaryotic protein, modified, moved, whatever, until it is functional, i.e. it's able to do its job. And so one of my favorite examples in terms of proteins is insulin, right? So I actually raided the Nutella jar this morning. Damn it, my wife's here. Didn't hear that, did you, love? No, she's in the pantry. And when you do have breakfast or whatever, raid the Nutella jar, your blood sugar level spikes, right? And so in response, your beta cells in your pancreas, which produce insulin, release that insulin, right? They don't make it in response to elevated blood sugar level. They release it, right? They store it in vesicles. And so that insulin is only functional when it is in your bloodstream and able to bind to the insulin receptors on your muscle and liver cells, main uptakers of glucose. In response, those cells take up glucose, blood sugar level drops back down to within its normal range, and you're good to go, right? The insulin as a protein looks nothing like what is actually produced by translation, because it's kind of chopped a couple of different places, like shooting. It's stuffed into what's called a vesicle, which is a small membrane-bound storage container. And it's kept there until the beta cells get a signal to release it in response to high blood sugar levels. right? So until that point, that protein is not functional. It cannot do its job because it is not able to bind to its receptor. And so there's a lot of steps that kind of go on, and that's why beta cells store insulin, right? Because they can't make it quick enough in response to that elevated blood sugar level. They have to make it kind of in their downtime, essentially. And that's also why one aspect of diabetes is kind of overwork of your beta cells, right? So if you have a constantly elevated blood sugar level, your beta cells are just running like flat out constantly to try and produce enough insulin, and they're like, screw this, I'm done, and they die. And so that's part of diabetes. It's not all of it. There's a lot of other aspects to it, but that's part of the reason why people get diabetes. Anyway, so that's what a protein is. Proteins aren't the only things that do stuff, though. RNAs do as well. So you have two types of RNA, largely. One, coding RNA, i.e. mRNA. Oops. And that's used to make protein, right? So that contains the information necessary to create that polypeptide sequence that I referenced further up in the chat. And then two, non-coding RNAs, of which there are many, right? And so those do stuff, but they do so themselves, right? They're not acting as an intermediate between DNA and protein. They are the functional outcome of expression of that gene. And some of them are here. mRNA is obviously a coding RNA, and all these other ones are non-coding RNAs, which is, RNAs are pretty funky things, actually. And so generally speaking, there are a bunch of steps in this process, right? So we have our DNA. We need to turn the information in DNA, because DNA is a static, well, it's not static, it's dynamic, but, it doesn't do anything of itself, right? Got to turn that information in DNA into another form of information, that's mRNA, or a non-coding RNA. And that information then has to get used to make protein through the process of translation. So that's a simple version, right? So transcription is a synthesis of mRNA, or RNA in general, by an enzyme called RNA polymerase. In prokaryotes, as soon as that mRNA starts being made, it can be translated because they don't have a nucleus. In eukaryotes, that RNA has to be processed via series of steps called RNA processing, right? And then that RNA, or mRNA, is used as the information with which the ribosome makes the protein. Now, that isn't necessarily the last step. There's lots of other kind of events after that, potentially, depending on the protein, right? It's very much protein specific and organism specific too. What happens to that protein, whether it's functional as soon as it's made, or if it needs a whole bunch of other stuff doing to it, like insulin, for example, before it is able to be functional. Oh, there goes the cat. And so one of the things I've banged on about a lot, right, is the fact that DNA is directional. Oh, sorry. Need my coffee. Right, it has a direction, right? And that's the whole 5' to 3' deal, right? So you can only make DNA in that direction by adding nucleotides to the 3' end. Hello, girl. Same goes for RNA. You can only synthesize it by adding RNA nucleotides to the 3' end. Sorry, scratching my dog as I talk. And also, because mRNA has directionality, so does protein. So protein has what's called an N-terminus and a C-terminus. And the N-terminus is the start. It's the amino end. And the C-terminus is the carboxyl or carboxy. Right, that's the COOH that you see in the diagram in front of you. Oh, now we have another dog. Ta-da! Polypeptide, Caitlin, is the sequence of amino acids that makes up a protein. So amino acid equals one unit. When amino acids get joined together, they equal peptides. Lots equals polypeptide. So it's a sequence of amino acids that makes up the protein. It's like the long chain, the linear chain of amino acids. That's a polypeptide. Does that make sense? Where do we cover that? Let's see. So basically, yeah, let's see if we can see what I'm looking at. Here we go. So I should do it the other way around, really, shouldn't I? Given that I'm right-handed. That is an amino acid. And the R group is what makes it a particular amino acid. Right, this part here, what color is this? I don't want that one. Purple, fine. Right, this part here, the NHH, that's the amino group. This here, this is your carboxyl group. Spell it wrong further up. Right, so that's the structure of an amino acid, right? Does that make sense? So because mRNA is read this way, oops. Right, so this is the direction of the information. That's all right. The next amino acid gets added here, right? So we have this one comes in. This is essentially what the ribosome and the ribosomal RNA do. Right, so here's the next amino acid. We remove water, and then we generate a peptide bond plus water. Right, so this is a dipeptide because there's only two of them, but then you move on and you make another one, or you add another one. So the next one would be added here, for example, right? Run out of space on the board. And so you just keep going along the mRNA, and at every time you get a piece of information, it's called a codon, that tells you what the next amino acid needs to be, or tells the ribosome. Amino acid comes in, peptide bonds formed, ribosome moves on to the next one, next amino acid comes in, and as you go along, now you can see you have this chain of amino acids, right? So here's another amino acid. Well, actually, technically, this is now a peptide because it's been added to another amino acid. And so you do that enough, now you're going to have potentially a polypeptide of thousands of peptides. That then gets folded up into whatever three-dimensional structure it, will end up assuming, and then it is a protein. So the order of events is amino acid, polypeptide, protein. Typically, we don't refer to proteins as polypeptides because they have other stuff done to them, whether it's folding or processing or whatever, that turns it from just a linear chain of amino acids into a three-dimensional functional product. Help? Should better put these back on because I can't see whether it did or not. Cool, good. Okay, so essentially that's the root of the directionality. Now the other thing which is kind of interesting about DNA, and this is something that takes a little bit to get your head around, is that DNA can only be made in the 5' to 3' direction, right? We know that. So can mRNA, or any RNA for that matter. So if the top strand, right, because it's going from left to right, is the coding strand, right? So this is the strand of DNA that encodes the information for whatever product is going to be produced, be it RNA or protein. The mRNA copy cannot be made using the coding strand because it's going in the same direction, right? So how would you make that? Right? You will always know that everything DNA and RNA is made in an antisense direction, right? So if you have what you want to make, you have to make the copy in that direction, right? So instead, what's done is that the other strand, the one that doesn't contain the information, that's used to make the mRNA. That's called the template strand. Right, so that's something that kind of takes a little bit to get your head around. We'll come back to it again in the future. Now, one of the consequences of that, however, is that because DNA has 2 strands, it can have 2 coding strands. Right, so both strands can encode information for the production of something. Right? And so this is the double helix, right? So we've got our, anti-parallel double helix like so. And essentially, you can have genes going in different directions. So you can have 2 genes going in this direction. So this is actually the template strand here for this gene. This is the template strand for this gene. And the actual coding strand is this strand. For the transcript being produced here, this is the coding strand, this is the template strand. Right, so you can have, and they can actually overlap. It's a little rare, but it's not unheard of for genes to be kind of actually overlapping in terms of their information, right? So one coding strand, the gene like so, the other coding strand like so, and they actually can overlap, which is kind of weird to think about. Other times, and in the case of some of my own work, I've actually found genes that are differentially expressed, right? So they're kind of expressed under particular conditions, right? Actually sharing a region of DNA. So in my case, they're actually like so, or like that maybe. And so there's a region of DNA in between, which presumably is used to control both of them at the same time, which is super interesting. Now, one of the key differences between prokaryotes and eukaryotes is that prokaryotes, the product, protein typically, is collinear with the gene, right? And so this might be like, well, duh, yeah, of course it is, right? That makes sense. But it's worth emphasizing because that's not the case with eukaryotes. And so in prokaryotes, whatever the DNA sequence is, the protein sequence maps directly to that, right? So if you have a mutation here in the DNA, you'll have a mutation in the matching, or a change, sorry, in the matching place in the protein, and so on and so forth, right? And you're probably looking at this thinking, okay, right? That's just like what you've been telling me all along, right? Yes, but when you look at eukaryotes, now they are not collinear. They are interrupted. Gene interrupted. It's like the new film. And there's a whole bunch of extra stuff. And so what we mean by interrupted is that we have exons and introns. Exons code for protein. Introns do not and have to be removed before translation. So these introns are essentially like spacers. They don't encode protein. They do have a functional purpose, although it's not always entirely clear what that purpose is. And every eukaryotic gene has at least one intron. If I can spell eukaryotic. Meiotic. And in terms of coding regions, right, the actual part of a gene which encodes the protein, eukaryotes and prokaryotes are pretty similar. It's around average, at least, is about 3,000 base pairs, give or take. Some genes are bigger, some genes are smaller. But the actual amount of DNA that a gene occupies in eukaryotes is about 3,000 base pairs, right? Give or take, on average. In eukaryotes, it can be up to 80, 90,000 base pairs. So 80 to 90 kilobases. And the sole reason for that difference are introns. Introns can be huge, tens of thousands of base pairs long, potentially not containing anything, right? It's kind of weird. They are kind of odd. Also, in addition to that, eukaryotic genes, or genes have what are called UTRs. And so UTR is an untranslated region. At the beginning, that's the 5' and end 3' of the gene. Well, didn't actually, of the mRNA. These are really important for mRNA stability, translation, and other stuff. Really, really important. So even though they don't encode protein, they are very, very important for the production of protein, right? So eukaryotic genes are interrupted, and we become coming back to this a bunch because obviously there's going to be a lot of stuff that needs removing, right? These introns will have to come out before translation occurs. Has anybody actually got any questions so far? Anything that needs clarifying or expanding upon? Cool. Yeah, and that's kind of what I said. Cat is back. Okay, so that's kind of the overview. Now we're going to get into the weeds, essentially. And so transcription is the process of making the RNA molecule using the information in DNA, right? So it's complementary to the sequence encoded by a gene, as in it is the same as, right? However, RNA differs from DNA in a whole bunch of ways. There's a, Chasnitz, where you go? What do you want? There are some obvious ways, right? So the obvious is uracil, right, instead of thymine. And also an extra OH in the ribose. That's why RNA is RNA, right? And it sounds kind of obvious, but it's funny to think about it, right? RNA is ribonucleic acid. Equals 2 lose my voice. Hydroxyl groups. DNA is the oxy ribonucleic, oops, if I can spell it right, acid. Dang it, don't correct that. Bloody spell check. Equals only one hydroxyl group. Now that lack of the second hydroxyl group actually benefits DNA in some ways. It makes it a bit more stable. But it also means that it can't do some of the funky stuff that RNA does. And RNA is a funky molecule, right? As funky as molecules can be. Let's see if I can find some cool images. Yeah, I don't need to know that. Ooh, that's pretty cool. RNA structures of coronavirus. Yeah, how about that? Super cool. Well, there you go. It's kind of like a pretty pertinent, relevant topic. So this is what the RNA genome of SARS-CoV-2 actually looks like. So you can see that it is weird looking, right? And it forms all of these kind of funky shapes, basically. Here's some other ones. I don't know where these are all from. And me. HCV is a type of virus, I think, off the top of my head. Cricket paralysis virus. It's kind of sad. I don't want to be a paralyzed cricket. Anyway, you can see that RNA forms like crazy, crazy structures. So DNA is pretty mundane in comparison, to be honest. Right? And these structures are really important for the function of that mRNA and its stability as well. And so that's, in my mind, actually a far more interesting difference between DNA and mRNA, so, or RNA in general. RNA can form really, really cool structures. Oh, also another difference. let me just add this. I'll upload this new one. RNA can act as an enzyme, and that's called a ribozyme, as well as store information. There you go. This is like teaching as it goes. So that's actually one of the kind of most interesting things about RNA. Anyway, we're not going to get into that too much here. We're really more concerned with it in terms of an intermediate conveyor of information, right? Which is really the purpose of RNA. And so RNA is synthesized very much in a similar way to DNA, right? It uses a DNA template to add the correct, complementary or opposite base. It does not require a prime because RNA polymerase does not require that. If you remember actually, here you go. What's the enzyme that makes the RNA primer and DNA replication in bacteria? Anybody remember that? Begins with a P. What's the general rule about enzymes? You take the thing that it makes and add an A's. Yeah, it's primase. Right, so primase is a type of RNA polymerase. And actually in eukaryotes, DNA pol alpha contains 2 primase subunits, right, in its kind of general ultrastructure. Right, so it doesn't require a primer, and the enzyme that makes it is RNA polymerase. So it looks pretty much like DNA replication, right, or DNA synthesis. You have this nucleotide coming in with a triphosphate. The energy in that are base pairs, right, based on size and hydrogen bonds, right, shape essentially. And then the energy in that phosphate bond here is used to create the phosphodiester bond like so, releasing pyrophosphate, right? And that just continues until it's told to stop, essentially. Now, there are a few different RNA polymerases. It's an enzyme which is huge. So it's a multi-subunit complex. And what that means, let me just type it out. What that means is it's one protein complex made up of lots of separate proteins. So it's not one protein alone, it's lots of different proteins kind of glommed together to create the right shape and to have the function of that enzyme. So in prokaryotes, they only have one. It contains 6 different proteins. In eukaryotes, there are 3, right, which make different things. So RNA pol 2 is the main one which synthesizes all protein coding genes. Right. So whenever we're talking about transcription in terms of gene expression, typically we're talking about RNA pol 2. It also makes these small nuclear RNAs. Those are involved in RNA processing. We'll come to that kind of later on. RNA pol 1 makes ribosomal RNA, and RNA pol 3 makes tRNA. So T is for transfer, that's involved in translation as well, and the small little bit of the ribosomal RNA. But the main workhorse, in a sense, is RNA pol 2. And it looks something like so. And so it has, and this is also pretty neat as well. So here's the kind of multi-protein protein, right? The RNA polymerase. And the DNA comes in, and because of the shape of the RNA polymerase, it bends the DNA. Now you remember from DNA replication, the way in which the DNA is opened up for helicase and single strand binding proteins to enter is it's bent around and it pops open the double helix, a weak point essentially, right, where there's lots of A's and T's. It's pretty much the same principle at work here. So RNA polymerase physically bends the DNA as it comes in, and in doing so, that pops open in a very temporary manner, right? Just for the bit that it needs. It pops open the double helix, which allows it to then use that as a template to make RNA. I wonder if I can find. There you go. It's actually a fairly, at least transcription itself is a fairly simple process. And so essentially you have, and there's not a huge amount of space here, this is just for simplification's purposes. You have a bunch of proteins which recognize where RNA polymerase should bind. And so the initiation of transcription is by far the most complex involved part of the process, right? Hey, leave it alone. So we have a bunch of proteins which basically kind of hang there and go, come on, RNA polymerase, bind me. And they flap around in the breeze until RNA polymerase comes and binds it, right? And it kind of assembles, again, that holoenzyme, right? And there's a whole bunch of different proteins kind of that assemble. And then what you're seeing here is a protein, a transcriptional activator protein bound to another bit of DNA. This is in a eukaryote that then activates RNA polymerase. Doink, and then off it goes. Off of the races. That's about it, really. So let's see, there's a kind of, let's see if I can slow this down. There we go. So you can see here is that bubble in the DNA where it's been popped open. You can see the bend as the DNA is bent around, and this is the new RNA strand being kind of extruded from the RNA polymerase. So coming in one end is the DNA as a double helix. You can actually see very nicely, actually, the minor and the major groove. That goes in, gets bent, and you can also see this flow of the yellow bits. Obviously, they don't have color, but the yellow bits coming in are the RNA nucleotides, which are being added to that growing RNA chain. Zoom. Super exciting. And really, it just kind of keeps doing that until it doesn't. And there's a bunch of different ways in which that's controlled too. So the actual kind of making of the RNA is in many ways the least interesting part of transcription, right? Or the least involved or exciting part, I guess. I'll add this to, let's add it to the chat, but I'll add it to the PowerPoint too, because that'll be useful. Let's get back there. Okay, so essentially what, and this is in prokaryotes because RNA pol 2. Now, essentially there's a lot going on, as I said, in terms of the initiation of transcription, right? And this relies a whole bunch on particular sequences in the promoter of the gene. Let's see if I can. There you go. So this is in eukaryotes, but it kind of applies to prokaryotes too, just in a slightly different way. So essentially, each gene consists of two parts. Right, I'll go back to the other stuff next class. So we have a transcription start point, right? So that's the point at which RNA begins to be made. So this contains the coding region. This is the bit that contains the information that's going to be turned into RNA or mRNA, and then potentially turned into protein, right? So we have the coding region of the gene, but we also have a whole nother region, which in eukaryotes can be really huge, which is involved in the initiation of transcription and the control of the initiation of transcription, right? And so this is called a promoter because it promotes gene expression. And that's the part of the gene that is involved in RNA pol binding and initiation of transcription. So essentially in that region, you're going to find DNA sequences that are involved in binding RNA polymerase, regions which are involved in whether or not that RNA polymerase actually starts transcribing, and particular parts which are, I'd say, weaker, right? So here's one of them, it's the Tata box, and you also see the same in, yeah, where are we at? Here we go. Bacteria, where you tend to find regions of high AT%, so high number of adenines and thymines. These are the regions which are actually popped open, right, to initiate transcription. And I think that's probably enough for today, to be honest. It's a Friday. Hey, free minute. And we'll get diving into this on Monday. All right, that's it from me. Does anybody have any questions before we sign off? My question is, how quickly can I make myself a cup of coffee? Take the dogs for a walk. Cool. All right. Well, same to you, everybody. Have a lovely weekend. Enjoy the beautiful weather we're having now. And I shall see you all next week. Bye.